Better Alzheimer's treatment soon. Alzheimer's is a crippling disease that currently has no cure, but a British neuroscientist is hoping that breakthroughs in research could lead to better treatment in just 10 years. According to a neuroscientist, Joseph Jabali, memory loss comes with age, but forgetting what certain everyday things are could signal a more serious problem. Healthy brains shrink by about 10% between the ages 50 and 80. Brain cells shrivel up slightly, reducing contact between each cell and causing them to function more slowly. In patients with Alzheimer's, toxic proteins cause a buildup of plaques and tangles in the brain, which causes cell death. Current medication only treats the symptoms and can delay them for 6 months to a year. Researchers have been looking into ways of using sleep to treat the disease, since the brain uses cerebrospinal fluid to clean away the plaques and tangles during sleep. Scientists are also considering neural stem cells as a treatment for Alzheimer's, believing that the cells can be activated to generate new neurons, allowing the brain to heal itself. Jabali believes the future of Alzheimer's treatment lies not in an outright cure, but in being able to manage and control the disease enough to keep the symptoms at bay. There's hope for Alzheimer's patients yet. New drug offers hope in fight against Alzheimer's. The drug aducanumab is believed to aid in the clearing of toxic proteins hypothesized to cause Alzheimer's disease. A recent study measured brain activity of 165 participants, some of whom received the drug once a month for up to 54 weeks, while others received a placebo. Of those who received the drug, 103 experienced a reduction in the amount of tangled amyloid beta, the toxic proteins thought to trigger Alzheimer's, in their brains. For years, scientists have debated whether the buildup of amyloid beta protein causes memory loss and ultimately Alzheimer's disease. In a clinical study, researchers injected the drug aducanumab in early-stage Alzheimer's patients. 1 or 2 in every 1,000 of the antibodies enter the brain, where they latch on to wayward amyloid beta proteins. Researchers believe that other cells called microglia then arrive and clear the aberrant proteins from the brain. The trial's results were in favor of the amyloid hypothesis, which suggests that the elimination of the protein itself might alleviate the disease's symptoms. 2 larger Phase 3 trials are now in progress and plan to run until at least 2020.
Detecting Alzheimer's years before symptoms begin. Researchers at Washington University in St. Louis have developed a simple blood test that may be able to detect whether a person is developing Alzheimer's disease. Amyloid plaques, the buildup of amyloid beta proteins in the brain, start developing more than 15 years before the symptoms of Alzheimer's disease start to surface. At the moment, the only ways to monitor plaque buildup are through PET scans or spinal tap procedures. Researchers have developed a new blood test that can detect the amyloid beta buildup in the brain. The test measures the amounts of 3 amyloid subtypes, the peptides amyloid beta 38, 40, and 42. It has been found that the levels of amyloid beta 42 are consistently 10 to 15% lower than amyloid beta 40 in people with amyloid plaques in the brain. The blood test is said to have an accuracy rate of 89% over 20 blood samples. Researchers plan to expand the experiment to include 180 people. Sleep tight if you don't want to get Alzheimer's. A new study shows that even a single bad night's sleep could raise levels of a protein that may lead to clogs in the brain and result in dementia. Amyloid beta and tau are 2 proteins in the brain that are naturally produced but can cause clogs and tangles in the brain. The proteins are cleared away when the body enters slow-wave sleep. Experiments have shown amyloid levels increase by about 10%, with tau levels also increasing after slow-wave sleep is disrupted for just a few consecutive nights in a row. Amyloid beta protein levels in excess of 10% could cause amyloid plaques that lead to dementia. Meanwhile, the increase of tau could cause tangles in the brain that lead to Alzheimer's disease. More than 5 million Americans have Alzheimer's disease, and many more others are expected to develop the disease as the population ages. There is currently no cure or treatment for the disease.
Does Alzheimer's originate outside the brain? Scientists have long assumed that Alzheimer's starts in the brain, but new research suggests that might not be the case. Patients with Alzheimer's disease have abnormal deposits of amyloid beta protein in the brain, which smothers the cells and impairs nerve and memory function. Amyloid beta protein can be found in other organs apart from the brain, though because of the blood-brain barrier, the amyloid responsible for Alzheimer's was believed to only originate in the brain. When researchers used parabiosis to attach a normal mouse to one with high amyloid beta levels, the protein traveled to the normal mouse's brain, effectively causing the animal to contract Alzheimer's in just 12 months. Scientists suggest that as the blood-brain barrier weakens with age, more amyloid beta may infiltrate the brain and contribute to its deterioration. The findings indicate that future drugs might be able to halt the disease by targeting the toxic protein before it ever reaches the brain.
A breakthrough in Alzheimer's research. Researchers at MIT have discovered a new potential treatment for Alzheimer's disease that involves interfering with an enzyme that forms blockades in brain cells. Scientists have found that levels of HDAC2 enzymes are usually higher in people with Alzheimer's. Medical mice with the disease also have elevated levels of this enzyme. HDAC2 enzymes bind to genes called SP3, which in turn condenses chromatin and reduces the expression of some genes in the DNA. This results in the blockade of memory formation. Researchers isolated the particular section of HDAC2 that binds to SP3 and overproduced that fragment. These fragments bind to SP3, preventing it from binding to entire HDAC2 enzymes, thus allowing memory-linked genes to be expressed. Previous attempts at blocking HDAC2 would often trigger dangerous side effects by interfering with the production of red and white blood cells. The MIT method is the first procedure to have shown no interference with other enzymes. Researchers are investigating other ways to adapt the technique in human trials.